morning, everyone. Thanks again for coming, and welcome back to our seminar series here at the Academy. I'm really excited to introduce today's first speaker of the series this spring. We have Isaac Overcast with us. Isaac has a really diverse background in things like AI and computational theory, and he's been working with the Academy since about September on a biodiversity resilience modeling project here, and hopefully we're going to hear about some of that exciting stuff for Thriving California today. So with that, I will go ahead and bring Isaac up. Thanks.
Thanks, Cal. Hi, everybody. My name's Isaac. I'm a research scientist with the Islands 2030 project. And I'm going to talk to you about some of the work we're doing to try and understand biodiversity resilience, particularly social-ecological resilience. I think my mom is watching the live stream, so I just want to say hi, Mom. So I'm attached to the Islands 2030 project, and just as a reminder, the mission of the Islands 2030 project is to halt biodiversity loss and habitat degradation in 5 key tropical island archipelagos by 2030, while empowering and benefiting local communities. The focal island archipelagos are spread across the globe. There's the Galapagos, the Lesser Antilles, Gulf of Guinea Islands, Madagascar, and the Philippines. And so the question arises, why are we so interested in islands? Well, islands are small, closed systems, so they're kind of more tractable. They're biodiversity hotspots. They host tons of unique endemic species. They're vulnerable and historically exploited, and the social and ecological systems are at greater risk of collapse. You know, the threats to and stresses that humans impose on biodiversity are kind of diverse, from habitat fragmentation, biological invasions, ecological disturbance, and of course, global climate change. These are all important stressors on biodiversity, and they have huge impacts in island systems in particular. So the mission of the Academy is to regenerate the natural world through science, learning, and collaboration. And what I want to think about for a moment is, what is the feature of the natural world that we want to regenerate, okay? So is it the woolly mammoth? Do we want to bring back the woolly mammoth? I think there's some people that are trying to do this, but what I would say is, well, you can bring back the woolly mammoth, but in order for the woolly mammoth to thrive, you would also have to bring back the vegetation and the other predators and other kind of organisms that it shared its environment with it. So I think that focusing on one particular species maybe isn't the best path for regeneration. So maybe we can think about expanding regeneration to ecosystems like mangrove forests, which are important for stabilizing coastal ecosystems. And this is great. I think we should regenerate mangrove forests, but this is only kind of relevant to areas of the globe where there are mangrove forests already. And so I think we could step back a bit and think about what are some kind of global features of biodiversity that we could focus our regeneration efforts on. One might be species richness. We can look at just any kind of ecosystem and think about how many species there are, and maybe, if we regenerate the number of species in an environment, then that could be like one metric that one approach that we could take. So we could go out here to Golden Gate Park. If we wanted to, increase species richness in Golden Gate Park, we could go down to the store and buy some like Alaskan wildflower seeds and just throw the Alaskan wildflower seeds all over the place, and species richness in the park would explode. But maybe this isn't exactly what we want to kind of propagate. And so what I'm going to tell you about today, and what I'm going to propose, is that biodiversity resilience should be the fundamental target of regeneration. And so, what are some of the properties that I think make biodiversity resilience an important focus for regeneration? Well, it's a reliable indicator of ecosystem health. It's a globally relevant property. You can look at resilience in any ecosystem across the globe. It's measurable, and it's self-propagating. So resilient systems, resilience begets resilience. So what I'm going to talk to you about today is, what is biodiversity resilience? I've kind of painted this picture of why it's important, but let's think a little bit more about what biodiversity resilience is and how we might study it. Then I'll talk to you a bit about a model, how we can construct a model of biodiversity resilience so we can study it. And then I'm going to end by talking a little bit about some work that we're doing specifically related to the Islands Project, studying and promoting social ecological resilience in Madagascar. So what is biodiversity resilience? I will say that it's the ability of an ecosystem to withstand external stress without changing state. So, here's some of these examples of stresses that humans might impose on an environment, and a resilient ecosystem would be able to absorb more stress without collapsing, for example. So here's just kind of an example of this. We can look at these two kind of examples of forest systems, one intact on top in the blue and one heavily degraded. And so now if you look at this figure here on the left, and we can just, let's imagine that we can measure biodiversity resilience on the y-axis. So you can see that the intact forest is very resilient, and the degraded forest is low on the resilience, and time is going kind of to the right. It's forward to the right. And so now if we look at this red dashed line here and we imagine that there's a stress imposed on the system, a resilient ecosystem will retain its resilience, whereas a degraded ecosystem will crash in its resilience. So we can study biodiversity resilience with computer models. That's what I do. I'm a computational biologist. I develop models of how we think biodiversity works so that we can understand it better. And we can use these computer models to identify properties of resilient ecosystems. So that's really what we want to do. We want to look at the natural world and study as many properties as we can, try and figure out what makes an ecosystem resilient. So now, a lot of what I do is simulation-based machine learning approaches, so we can use these computer models of biodiversity resilience to kind of quantify resilience index. So we can assign a resilience index to an ecosystem. So if you look on the right here, you can imagine a kind of two-dimensional resilience space, I'll call it, where in the kind of lower left quadrant are vulnerable habitats, in the upper right-hand quadrant are resilient habitats. And so we can go out and sample a community and kind of plot it in this resilience space. And then we can use our model of how resilience works to predict recovery trajectories of different ecosystems under different conservation strategies. So you could see these two kind of cartoon examples of conservation strategies, protecting habitat or mitigating invasives. And we could kind of use our trained machine learning model to predict where the ecosystem will go under these different remediation strategies. And then, of course, we could do this across entire island systems. We could go sample and understand how resilience is distributed across the landscape in order to inform our conservation strategies. So how do we measure resilience? And what I'm going to propose to you is that we can measure biodiversity resilience using genetic data. And so this is real data from Hawaiian arthropods. So if we just look at Kauai and Hawaii, Hawaii being the youngest island and Kauai being the oldest, we can sample arthropod communities from these islands and sequence their DNA and look at the distribution of DNA and of genetic variation in the community. And so something that we know about Kauai is that it's older. The communities are more robust. They're more resistant to invasion. They're more resilient to human disturbance. And in the Big Island, the communities are super invaded, and they display fewer properties of resilience. And so if we look at the distribution of genetic variation in these communities, which I'm going to show you a ranked genetic diversity plot, you can see that in Kauai, in general, there's more genetic variation. There's more genetic diversity in more lineages in Hawaii or in Kauai than in the Big Island. So you just in the Big Island, there's a few species that have lots of genetic diversity, but most stuff is like pretty genetically depauperate. So the reason that DNA sequence data is useful for kind of understanding biodiversity resilience is because sequence data records historical information. So if we look at, for example, this beetle species from Cyprus, you can sequence the DNA, you can sequence the whole genome or REDS, a reduced representation of the genome, and you can reconstruct the history of the population size through time. So in here again, the time is on the x-axis and it's going from present on the left to the past, and the position of the line is showing the population size kind of backwards in time. And so we can use this information in the DNA sequence data to kind of understand how past processes have impacted this particular species. So for example, where there's this kind of precipitous drop in effective population size around 1,000 years ago, maybe we can correlate this with some kind of human habitat like land use change or something. I don't know. Maybe a lot of the times we're thinking about like the last glacial maximum or about human arrival on islands and creating disturbance or stuff like that. So the historical information in DNA sequence data is super useful for us to understand the past processes that have generated biodiversity patterns, and then we can use that information to predict how change will happen in the future. So if we look at just one individual organism, that's great, we can get a lot of information, but maybe this particular beetle is not representative of kind of the response of the community to past change. Maybe because of life history or because of just some random events, this particular species' history might not be representative. So what we need is to sequence the DNA of the entire community, of kind of everything across the landscape. And so how do we do this? We go out and collect the beetles from kind of all of the forests in this region and sequence everything. And so this is what metabarcoding, the metabarcoding approach is really about. So this could be like sampling environmental DNA or soil, for example. So you can go out to a site and take a soil sample and bring this back to the lab, extract DNA. Then you can amplify DNA sequences which are associated with a marker like CO1 or something like that, and then sequence all of the DNA in your sample, and you get this representation of the kind of genetic structure of the community. You can understand species richness, relative abundances, and then most importantly, you can get genetic diversity at the kind of ecosystem scale. I'd love to talk to you more about this, about metabarcoding and eDNA and stuff, but if you want to go kind of dig deeper, there's this TED Ed lesson that I helped to write when I was in my previous postdoc in Paris, and there's also a special issue of Molecular Ecology where we're talking specifically about how we can use metabarcoding approaches to understand ecological and evolutionary processes, which I co-edited, which just came out a couple of months ago in Molecular Ecology. So go look at those. So let's say we have metabarcoding data. Now, how do we use DNA sequence data at the community scale in order to infer ecological and evolutionary processes? How do we understand and predict biodiversity dynamics using this I'll call this biodiversity genetics data? So a couple of years ago, I formed this working group with experts in all different kind of domains of ecological and evolutionary theory. And we sat down and tried to kind of sketch out an answer to this question. How do we use DNA sequence data at the community scale to understand biodiversity processes? And so, what we kind of came up with is that if you want to understand how biodiversity works, you need to have a model of all of the processes that are kind of relevant to biodiversity. So one process is, some of the processes that are relevant are like speciation and extinction. These are the processes that generate new organisms. Another process is community assembly, where individuals are born and they die and they disperse between different habitat patches. And another kind of suite of processes related to population genetic variation, so drift and genetic mutations. And so what we kind of are proposing is that in order to understand how biodiversity works, we really need a unified model to kind of encapsulate all of these processes simultaneously. And so that's kind of the core of what my research is to develop unified models of biodiversity that account for processes across all hierarchical levels of organization. So I'm going to tell you a little bit about one of these modeling approaches. But so let's return to the beetle communities in Cyprus for a moment. So my collaborators, Anna Papadopoulou and Victor Nogorales, were interested in understanding what the processes are that shape soil arthropod communities in the montane forests in Cyprus. So Victor went out to the forest, went out to the mountains, and he sampled a bunch of arthropod communities. And so this is two kind of lenses on the region that he was sampling from. There's an elevation map, and on the top and on the bottom is a figure that shows the distribution of different habitat types. So in this case, there's 4 different habitats, but there's kind of 2 general features of these habitat types. There's 2 kind of general categories of habitat type. There's the widely distributed pine forests in the light green, and the oak forest in the orange, which also is kind of widely distributed. So these 2 habitat types, which are kind of broadly distributed throughout the mountains. And then you have in blue, kind of on the left, there's this patch of cedar forests, isolated patch of cedar forests, and then the high elevation pine juniper forest in black and purple. So Victor went out to each of these sites, and in each of these sites, he collected soil and extracted the arthropods from the soil and sequenced the bulk arthropod samples, like this. So each of these points, he has genetic data for all of the arthropods in his sample. And so what Victor found, just using kind of traditional community ecology methods for evaluating community structure, is that the arthropod communities are kind of associated with different habitat types have broadly different arthropod communities associated with them. So you can see that in this figure, where each of the points on the figure is a community, and they've been kind of collapsed into this 2-dimensional space where proximity of a point to other points is kind of indicative of similarity of the communities. And so you can see, for example, in orange, all of the oak forests are like pretty close together in this 2-dimensional space. And the pine juniper forests are all kind of close together in this space. So Victor found that the habitat type was very important for structuring these arthropod communities, but his approach wouldn't allow him to address the question of why. So why? What is the process that is driving this difference? Is it just neutral variation, just kind of organisms just happening to get to disperse around the environment, and you have this pattern, or is there some kind of competition or environmental filtering that's driving these differences in the community? So this is the kind of question that I'm interested in addressing with my modeling approaches. And so I'm going to tell you just a little bit about the MESS model. This is the Massive Eco-Evolutionary Synthesis Simulations model. We have this little logo on the bottom. I have stickers. I have stickers. So if you want a MESS sticker, I forgot to bring them, but you can come downstairs and I'll give you one. I have them here. But essentially, we're not going to go through all of this stuff, but what I want you to kind of see at a high level is that the organization of MESS is exactly what I told you before. There's a kind of larger scale, deep time speciation extinction process. There's local community assembly dynamics with individuals being born and dying and dispersing. And there's a population genetic process that's associated with that. So we can, yeah, so essentially it's one model to encompass all of the processes, many of the processes that we think are relevant to how biodiversity works. So I'm just going to give you like a little kind of taste of how this works. I don't want to get kind of lost in the modeling details, but we can return to this intact forest, and I'm going to show you how we kind of model this, how just the kind of intuition for how the model works. So we can take this snapshot of the forest and kind of convert it into this this kind of computer representation where we have an isolated patch of habitat, which is a fixed size, a fixed number of individuals. And so let's think for a second, what are the kind of, if I tell you that the model kind of proceeds in generations, like we can model one generation of trees by another, what are some of the processes that we might consider to be relevant? Individuals may, I'll give you a hint. Yeah, so individuals can die, thank you. Individuals can die, that happens, you go out into the forest, individuals can die. New individuals can be born to take up the ecological space of the dead individual, and stuff can come in from outside, right? So new stuff can come in to Golden Gate Park from somewhere else and establish. And so really that's all this model really is it's a model where organisms they die and they're born and they get replaced by something from the local environment, or they get replaced by something that comes in from outside. So we can just run this model in order to kind of simulate community biodiversity dynamics. So this is really, I didn't invent this, this is Hubble. So this is the unified neutral theory of biodiversity. And what makes this particular model neutral is that all of the organisms in the local environment are ecologically exchangeable. So there's no fitness difference, there's no kind of difference in probability of being born or of dying that's related to species identity. And this model makes surprisingly kind of coherent predictions for distributions of abundance in communities, but people don't like it because they don't believe that ecology is neutral. And so we use this kind of classic model as a foundation for MESS, and we incorporate a couple of different processes into it in order to give it more kind of biological relevance. And so one of those kind of sets of processes is we actually kind of infuse the model with ecology. So we give organisms traits, and we allow the traits to interact with the environment, or we allow the organisms to interact with one another based on traits. So there can be an environmental filtering process imposed where the environment has some characteristic that can be either matched or not matched by the traits of the organisms that can confer some increased probability of being born or of dying or of establishing. We can also do something similar with the trait values where organisms compete with one another for space. And then we can also incorporate a process of local speciation. So there actually can be kind of novel species arising in the local community, which is not kind of a feature of the original Hubble model. And then on top of all of this, we do some population genetics black magic, but I'm not going to kind of get lost in the details of that. And so all of these processes are kind of controlled by parameters. So we can manipulate the parameters of the model in order to look at how different parameters change the predicted outcomes in biodiversity, in patterns of biodiversity data. So we can turn competition all the way off or all the way on. We can turn speciation all the way off or all the way on. We can kind of tune, we can explore the behavior of the model for a variety of parameter configurations. Yeah, so MESS is like, as a unified model of how biodiversity works, it makes joint predictions of species abundance and genetic diversity, phylogenetic relationships, and trait values. And we can use this information to fit the model to empirical data. We can actually go out and sample data from empirical communities, come back and do a bunch of simulations, and we can kind of use machine learning to find the parameter values that generate predictions of biodiversity patterns that are most like what we see in nature. And then we can infer something about the process from the parameters that generated those predictions. So I'm just going to show you a little kind of taste of how this works. So for this kind of operation, what we're going to just imagine is that all we care about is these three kind of like broadly different community assembly processes. We care about, we'll just imagine environmental filtering, ecological neutrality, and competition. So we're just going to simulate different biodiversity genetic patterns. What we want to do is we want to evaluate kind of grossly how these different assembly models generate different predictions of biodiversity genetic patterns, just kind of like at a broad scale. So how does this work? Well, we take each of these models and we simulate, 10,000 or 100,000 different realizations of, because it's a stochastic model, so each simulation kind of is slightly unpredictable. And so in order to understand how the model works, we have to generate a lot of simulations and kind of like squint at the patterns of data. So we do all these simulations, and then we can train a machine learning model on the simulations. We can train a machine learning model to differentiate between the different patterns in the data and classify the community assembly model based on patterns of the genetic data. Then we can go out and we can sample an empirical community, and we can interrogate the observed empirical biodiversity genetic data with the AI model, with the trained AI model, to make predictions about the community assembly dynamics that generated the observed patterns. So in this case, this empirical data is classified as being primarily neutral. Just a cartoon example. So now let's return to Victor's question about the soil arthropods in Cyprus. So I'm going to give you a hint about the predictions of the model. So and I've kind of telegraphed this from the beginning, but the widely distributed pine and oak forests, these kind of green and orange dots, they were classified to have the same assembly model, and the narrowly distributed habitats were classified also to have the same model. And so I want you to think for a second, broadly speaking, what community assembly dynamics you would expect to obtain in these kind of like widely distributed habitats. Who wants to guess? I heard neutral. I don't know who it was. Neutral. Very good. Okay, and now in the narrowly distributed habitats, what's these environmental filtering? That's exactly right. So this is like really kind of, I've done this many times, and everybody always gets it. So this, I think, is like a good example of how our intuition about how biodiversity works is kind of recapitulated by the model. So this is just kind of an advertisement for some of the work that the role model was kind of continuation of the MESS. The work that we were doing in MESS, this project funded my previous postdoc. It was led by Andy Rominger at the University of Maine. And so in this project, this is still ongoing. It kind of got put on hold a little bit by COVID, so we're still collecting data. But essentially what we want to do, we want to collaborate with empirical biologists across geographic and taxonomic domains in order to apply the model to these different systems to see, well, is this result we got in Cyprus kind of like an outlier, or is this like a general feature of how biodiversity works? So stay tuned. We're still working on this. Okay, so this MESS, I've showed you, what about biodiversity resilience and how we can kind of quantify it and use it, and I've showed you this model which this ecological and evolutionary model, which we can use to understand as kind of like a springboard for understanding biodiversity resilience. And so now I'm going to tell you about how we can use this in an applied context, this kind of like modeling approach and biodiversity resilience in an applied context. So in November, myself and several other folks at the Academy wrote this and submitted a grant to the NSF DICES project, which is the Dynamics of Integrated Social-Ecological Environmental Systems. And this was the title of the grant, using transdisciplinary community-based participatory research to understand island social ecological resilience. And so I'm going to give just a special kind of shout out to our collaborators at Stanford in the Arduin lab, who were instrumental in kind of developing this project with us, and it definitely would not be what it is without their collaboration. And I think that this is really a wonderful example of how this kind of DICES program, which is about marrying biology and social science, social ecology, to try and understand the feedback between social and cultural and environmental systems. And so this is kind of the conceptual overview figure for our grant, where we have these kind of three overarching components. One is social ecology work, one is broad spectrum biodiversity surveys, and one is biodiversity resilience model. And so the concept of this grant really is to, and it's illustrated with this kind of figure here, where we want to like inform the biodiversity surveys with the social ecology work. We want to inform the biodiversity resilience model with the social ecology work and the biodiversity surveys. And we want that to feed back into kind of continuing iterations of this cycle, where we aren't just coming in and saying, this is how we think biodiversity works and this is what we think you should do. We're actually going into local communities, trying to understand how people locally conceive of biodiversity and what they value about it, and how, what they want to see for their environments in the future, and then using that to kind of parameterize the survey, the sampling design for the surveys, and use that information to kind of inform how we do the model development. So I'm just going to give you kind of like, this is all very aspirational part of the talk, because we haven't really done any of this yet, but we're proposing to work in Madagascar. So I'm going to show you kind of the broad outline of each of these three components of the grant. So this is the kind of social ecology component, where in the first phase, along with our collaborators at Stanford, we will go out into different parts of Madagascar and identify local communities that are proximal to intact and fragmented forests. And then we'll develop partnerships with these local communities and, work with them to understand local knowledge and document community priorities for conserving and promoting biodiversity resilience in their kind of surroundings. So then in the next phase, we take this data. This is all the stuff that the Stanford folks are going to do. We take this data and we kind of like process the results from the community listening sessions and bring them back to the community, and co-develop an understanding of what the results have to say. Again, it's kind of this continuous process of we're adopting a community-based participatory research kind of design here. So really, we want the communities in these different regions to be involved, not only in designing the research, but also in kind of interpreting the results and presenting the results to the broader community and to the scientific community. And then finally, we'll use the results from phase 2 to design scenarios for testing with the biodiversity resilience model. So essentially, we want to use the kind of community listening sessions to identify sites to sample and understand how community biodiversity priorities, and then also to develop models for biodiversity remediation and conservation that we can test with the kind of computational model. This I already showed you. We're proposing to use broad spectrum eDNA metabarcoding surveys, and yeah, just kind of again to reiterate the point, we're going to be sampling. We propose to sample in intact and fragmented habitats, and also in habitats that are culturally relevant, that are identified from community listening sessions, in order to kind of understand the spectrum of biodiversity resilience in that are proximal to our kind of community collaborators. And then this is the kind of third component of this project, is the biodiversity resilience model. So again, kind of aspirationally, what we're hoping to do is use MESS as kind of the foundation in order to, if we can quantify biodiversity resilience, then we can generate simulations that show how kind of resilience changes through time under different scenarios. We can do tons and tons of these simulations. Again, so on the top you can see this is just a cartoon kind of representation of, you can imagine the blue lines are resilient communities and the red lines are unresilient communities, and we can do tens of thousands or hundreds of thousands of simulations to kind of like train a machine learning model. This is the Island Social-Ecological Resilience, that's ISER. We can train the Island Social-Ecological Resilience model to kind of understand how biodiversity processes, how biodiversity resilience kind of processes through time. And then we can use that trained model in association with community identified conservation strategies. Like, these are just random things that, I'm using to kind of give you an idea of what we could, of potential conservation strategies. But of course, the conservation strategies we end up focusing on will be the ones that are most relevant to the local communities. But what you can see in this figure on the bottom is we can, for example, on the left-hand side of the vertical dashed line is this kind of biodiversity resilience trajectory, and the vertical dashed line represents kind of now. And then we can take these two different conservation strategies and forecast the biodiversity resilience in outcomes in the future based on these different conservation strategies. So just kind of reiterate this is something I think is really interesting about computer simulations and machine learning approaches, is that there's kind of two strategies that you can adopt. One is understanding how biodiversity has been structured in the past. You can use empirical data and kind of categorize or predict the importance of different processes or assembly dynamics from contemporary data. And you can also use such models to predict future trajectories given a model and different hypothesized remediation strategies. So kind of just to wrap up, there's all these kind of stresses that humans are imposing on biodiversity. And I think that, what I hope that I've kind of showed you today is that we can use biodiversity genetics, metabarcoding, and computer simulations, computer models of biodiversity, and artificial intelligence in order to kind of try and define remediation strategies, like understanding adaptive capacity in communities or understanding how meta-community dynamics kind of remediate fragmented habitats, understanding invasion resistance in communities, and again, understanding how kind of biodiversity resilience works so that we can support and regenerate biodiversity in the future. Thank you very much. Yes.
Got you.
So that was awesome. Really great talk. To understand resilience, one must look at resilience to a particular disturbance for a particular thing. So one characteristic of machine learning is that we don't know exactly how it figures out everything. In other words, it's a mess. So how do you test the model's predictive value for actual disturbances for actual things?
Yeah, that's a really great question. Of course, it's hard to, experiment on biodiversity in the wild, right? That's kind of difficult. But one thing that I have been discussing with other people is, potentially designing some kind of microcosm experiments or mesocosm experiments where, we haven't done this yet, but if we can observe that microbial communities kind of follow similar community assembly dynamics as macroscopic communities, then we can use kind of microbial mesocosm experiments, and we can induce different kinds of disturbance in these systems, and then we can have a kind of experimental validation but just kind of depends on how much you believe that how microbes work can be extrapolated to how kind of macroscopic stuff works. I'm always optimistic about this kind of thing, but maybe, it's not something that we've done yet. But that's a really, very important question for sure. How do we know it works?
Good answer. Going to follow up. What about using the historical records? So for example, the beetle example from Cyprus,
Yeah.
where it has that big drop somewhere around like 500 AD. You mentioned things like glaciation, deglacial cycles certainly imprinted those genetic histories. Something like 500 AD, the whole Eastern Mediterranean changed with the collapse of the Western Roman Empire. So, I guess my question for that specific example would be, are there other species that showed a similar impact to suggest that maybe it's not just, a stochastic thing for that one species, but maybe there was a broader impact in the arthropod assemblage? But certainly, some of the experiments that you suggested in response to Terrell's question have already occurred in the recent past with invasions and extirpations and so on, population reductions.
Yeah, that's a great observation. I don't know if did I cheat? Yeah. So there's this I only got to talk to you about a small kind of fraction of our work, but one of the things that we're hoping to do, which I hope kind of addresses your question, is use information from paleo records. So this is a project that I'm working on with some folks at Maine, where we want to study dynamics of alpine plant communities. And one of the collaborators, Jacqueline Gill, is an expert in plant community assembly and ancient DNA. And so one of the things that we want to do in this project is take soil cores from lake beds and then go through slices and do metabarcoding on the slices so that we can reconstruct the kind of historical plant community assembly dynamics and then feed all that into the model, right? If we can kind of use this historical information to kind of constrain the predictions of the model, that would be great. Yeah. I don't know if I exactly answered your question.
Okay, one in the back.
Thanks, Isaac. That was great. Have you thought about using successional data to train your models? So understanding recovery after fire, or people have temporal scales of the communities that have recovered through time based on different management strategies. Is that something that data is available for that you could use?
I mean, that's a wonderful idea. I'm not sure. Yeah, there's so much. There's so many people doing really great work trying to understand wildfires, and how like communities, how kind of like how we're managing forests and how that can kind of promote fires, and how how that the impact that has on biodiversity. I don't know that much about what kind of data is available for those kinds of systems. But I mean, it's something that I've been thinking about a lot. It's a really great observation for sure.
I was wondering if there was a reason you chose the soil arthropod community specifically. Are they good indicators of overall biodiversity, or that's what they were interested in?
That's also a great question. I mean, Victor and Anna are entomologists, so that's kind of why they chose the soil arthropods. And a lot of the work that I've been doing is with arthropod communities, because it's, from a kind of pragmatic perspective, it's kind of easier to sample an arthropod community than like a vertebrate community, for example. In terms of whether arthropod communities are like indicator, like a biodiversity indicator, I have no idea. But that's, I would love, I mean, one of the things that I think would be, that I'm hoping to get out of this DICES project as kind of like an auxiliary outcome is this, the eDNA data that we're proposing to gather is going to be broad spectrum focus. So we'll get arthropods, we'll get vertebrates, we'll get plants, we'll get microbes. And so then if we had enough samples, then we could, we could look like in a resilient system, are the arthropod, is the signal in just the arthropod data representative? Always. Does arthropod kind of community health correspond to kind of, biodiversity health in toto? Or does it not correspond, right? That would be super interesting to find out, but I don't know.
Any other questions?
So along those same lines, talking about scaling it up to the ecosystem, like for people who are interested in conserving populations or particular, rare taxa, do you think that something that could come out of this model would be, insights at that lower scale? Like, could you look at the genetics or the population structure of a particular group and say, oh, this one's going to do well, or this one is in trouble and we should conserve it?
He promised to give me a tough question and he did it. I mean, that is a good question because, the kind of focal unit of interest is the community. And so the specific species identity of any given species within the community is like not something that I've thought much about how to kind of dial into. Yeah, I mean, it kind of depends. I could imagine something where you could kind of inform the model with the biodiversity that's local to, let's say you have a rare species. I mean, the genetic data would tell you, like, is it rare because it's gone through a recent bottleneck, or is it rare because it's been rare for a long time? And so that could be some information that you could plug into the model, right? And then you could get expectations out of the model of like what the probability of persistence is of a organism that had the same kind of history. But I never have thought to try that yet, but it's a good idea.
Hi, great presentation. So as an ecologist, I know how much effort and resources go into getting, this data that you're using to inform your model. So I kind of just wanted to ask if you've thought about, I guess, like the longevity of this tool and how, like, if you're planning on having kind of like multiple sampling over multiple years in order to make this tool stay relevant as these communities are changing, especially considering how fast they might be changing?
Yeah, that's a great question. I can tell you, thinking aspirationally, what my kind of aspirational thinking is like the kind of, I don't know if this gets to your question exactly, but like, the process that I described of how you kind of do the simulations and then train the model and then do the empirical analysis, I think that that's somewhat limiting in how accessible the tool would be to kind of the ecologists broadly defined. So, one thing that I've always kind of had in the back of my mind is to, in kind of analogous to like a ChatGPT kind of thing where we could, if I could just get a supercomputer cluster and just simulate everything, I could imagine just kind of doing all of the potential possible simulations you could ever want to do, plugging these all into a predictive model, and then putting that up on the internet as like a web interface where you could just take whatever data you have and put it in and then do, get some biodiversity insight from this kind of pre-trained model. And then, I guess part of your question was about like temporal sampling. Yeah, I mean, that's relevant to kind of what we want to do here to kind of, the more information that you have, like if you have a sample from here and from here and from here in time, we can put that into the model to constrain the, to constrain it to only represent what we know, happened to kind of constrain the predictions. But that's a great getting temporal data can be hard, can be kind of hard.
My question kind of piggybacks from that. Have you guys or any of your collaborators worked with marine systems in particular? Because a lot of them are much more like temporally dynamic at like an interannual scale or
Yeah.
seasonally.
Yeah.
And so when you sample is going to make a huge difference on who's there.
Yeah, that's true.
More mess, extra mess.
Mike Dawson is one of the collaborators in the Roll Grant, and he does work in marine lakes in Palau. Yeah, but you know, I think that what your question is really getting at is something that keeps me up at night sometimes, particularly when I think about like the DICES project and eDNA sampling, and how the sample that you would get would be, you know, go out on one day and sample, you come back 6 months later, and the signal that you would get from that might be substantially different, right? Just for a whole host of reasons. And also this gets back to the question about the mesocosm or microcosm experiments. Like, you know, it's kind of about like, yeah, I don't know. I mean, what my hope is that for the DICES grant, if we can control for, you know, time of day and season, you know, if we can kind of control for that as much as possible, then we can, you know, be hopeful that we're kind of getting a representative sample, comparable samples. But like, yeah, I know that we can't go out in the dry season and the wet season and expect to get the same results. You know, if in an ideal world with an infinite amount of money, we just go out and set up like a little data logger and just sample every day. But that's not, we're not there yet.
Okay, well, thank you so much, Isaac. If you'd all join me in thanking Isaac.
Thanks, everyone.